Hi there, friends. So, we are continuing our discussion of bacteria metabolism, and what we're going to do today is talk a little bit more about the pathways and some interesting aspects of these pathways and what it means for bacteria. And then on Wednesday, we're going to get into some of the more conceptual, easier stuff in terms of enzyme control and just summarize some of the different things that bacteria can do with these metabolic pathways. But let's kind of have a, previously on microbiology, we were discussing, where'd my big pen go? We were discussing the mechanics of the breakdown of glucose, just as a pretty good example, as a place to start. Glucose is a pretty easy one to think about. And we were looking at where the carbon dioxide is coming out, and we were looking at where the ATP is being generated. That is, where the ADP is being rephosphorylated. And we finished up with this, or this is where we ended up. We have, first off, glycolysis. Glycolysis, we start out with the 6-carbon glucose, and we require 2 ATP to start it, and we're going to need 2 NAD to pick up electrons. And this is what's going in. Can you see that? Well, the word in is right there. All right. Now, what comes out is 2 3-carbon pyruvates, or pyruvic acids, 4 ATP, and 2 NADH. So we've got a net gain of 2 ATP. And I was looking at the book. The book talks about 38 ATP. I say 36 ATP, potato. Actually, the exact number of ATP that you generate from one molecule of glucose is a little bit tricky to actually put a number on. Partly, there are ATPs that are being used to move things around, and the efficiency of the last step, which we're about to get into, is not 100%. Let's not get quibbling over it. Let's just go ahead and say 36 ATP total. We've made 2 so far. Whoopee! Now, next comes Mr. Krebs cycle. The Krebs cycle. Now, what I do is I kind of put these 2 things together. There's what's called the oxidation of pyruvate, where we're going to take a carbon out and turn it into a 2-carbon acetate molecule, couple it to something called coenzyme A, and make acetyl-CoA. That's the official starting point of Krebs, but let's work with it this way. So what goes in is the 3-carbon pyruvate. Okay, that's not exactly correct, but for my purposes, let's work with it. It makes it easier to write up on the board. And then rather than putting a number on it, how about we just say lots of NAD is required, and FAD is required as well. And then what comes out is 3 carbon dioxides. We add electrons to all these NADs to generate NADH, and we make an FADH2 molecule, and we make 1 molecule of the nucleotide triphosphate GTP, which we're going to actually throw into our ATP count. All right, this is happening twice because we have 2 pyruvates. So now, if we do our ATP tally, we've got 2 ATPs net gain from glycolysis. We have 2 ATPs, GTPs, it's basically the same thing, generated in the Krebs cycle. That's 4 total. We're supposed to get 36. I'm missing 32. Where do they come from? Glad you asked. the last step. This is the Nobel Prize winning step. This is a step that took a long time to figure out because it truly is a really weird mechanism. I mean, it is not something that would be obvious. It took a lot of experimental work to do. And this last step does require a little bit of black or whiteboard space, so I'm going to erase this. This last step goes by a couple of different names as well. And, you know, one of the things with this whole pathway system is they have been discovered kind of in bits and pieces. They were discovered in part before what was exactly going on was understood. And so sometimes the names are not really very useful. In this case, I have a bone to pick with the names in this one. The pathway that we're now looking at is known by a couple of different terms. It's either known as oxidative phosphorylation, and that is not a very good term. Another part of the system is called the electron transport chain, and that's a better term, but it doesn't cover everything. And rather than diagramming it at first, what we're going to do is talk about so not so much what goes in, what comes out, but what's happening, what it does is an easy way to summarize it. And so we can say this. Oxidative phosphorylation, the electron transport chain, does a couple of really critical things. Number one, it's going to recycle and reoxidize NADH back to NAD, FADH2 back to FAD, and a really critical point is the electron has to go somewhere. Now, in you and I, and in a lot of bacteria, oxygen is the final electron acceptor. And so we can make a note here, O2 is the final electron acceptor. And it looks a little bit like this. An electron and O2 are going to combine. That is to say, oxygen is going to be reduced. It gains an electron, and it becomes a molecule of water, and we're left with the oxygen radical, or 1/2 an O2, that is going to get picked up by the next electron coming down the chain. So that's the number one thing that the electron transport chain is doing with oxygen anyway. And just as a little note, you've been talking about perhaps in anatomy and physiology, this is the only place in the entire metabolism of animals, that oxygen is used. In fact, I'm saying with bacteria, oxygen is used nowhere else. Critical for us to breathe, it is used only in this spot to pick up electrons. That's it. Now, we'll see actually how some bacteria can get around this and use a different atom to pick up electrons. They can actually operate without oxygen. And really all they're doing is they're using something like sulfur or iron here. Doesn't work as well, but they don't have to have oxygen. So keep that in mind. Now, that's all well and good, and I'm kind of running out of whiteboard space again, so I'm going to erase this. Could you just write it down? Or pause it and write it. Here, I'll pause for you. Get out of the way. Enough. Number two, and this is where I have another vocabulary bone to pick with science, but you know, what can I say? The second thing is the oxidative phosphorylation electron transport chain system is going to generate a hydrogen ion, or also known as a proton gradient. We're going to have hydrogen ion pumps pumping protons out into a different compartment. And the second part of this is we're going to drive ADP rephosphorylation by using the proton gradient. Now, let's kind of unpack that term for a moment. First, ADP rephosphorylation in the parlance, in the vocabulary, this is referred to as ATP synthesis. But that's not quite right, because we've got ADP floating around. ADP synthesis, or the synthesis of the nucleotide triphosphate or diphosphate, is a whole different pathway. The synthesis of the nucleotide is going on because you need the nucleotides for RNA and DNA. This is the rephosphorylation of ADP. And when we talk about this business, it is a giant recycling system. And for bacteria, for you, what the bacteria is doing is incorporating glucose, incorporating oxygen, and generating ATP from this. But we don't actually incorporate ATP or ADP. We are simply taking that pool of nucleotide, and it's either in the triphosphate form, yay, or it's in the diphosphate form, I'm hungry. That's all you're doing, is simply going back and forth. In a human, the average ATP molecule is phosphorylated, dephosphorylated, and then rephosphorylated something like 500 times a day. Additionally, if you were to calculate up the amount of ATP that you're phosphorylating, that you're generating, it would be equal to about half your body weight every day. Now, obviously, we don't eat it. We don't increase our body weight and then lose it. It's because we're simply recycling it. It's just an important thing to point out. And so we call this thing ATP synthesis. And in fact, the enzyme that does it is called ATP synthase. But it's like, no, it is the ADP rephosphorylator. Thank you. Oh, well, that's my little, rant for the day. I'm done now. All right. So let's look at how this system works from a graphical perspective. Now, if you recall, in our endosymbiotic theory mechanism, we have proposed that bacteria-type cells have been taken up, engulfed by a primitive prokaryotic cell. And this is what we know as the mitochondria. Now, the bacteria itself doesn't have mitochondria. There are no organelles in a prokaryote. So they do things a little bit differently than what a mitochondria might do. But the system is basically the same. So let's look a little bit at what is actually happening here in this electron transport chain. So it goes like this. Now what I'm going to give to you is more or less the specifics to what's happening in the mitochondria, but it's also happening in the same way in bacteria. The names have been changed to protect the innocent. There's a little bit of differences, but let's not worry about that so much. All right, so we've got a membrane, a phospholipid bilayer. Now this phospholipid bilayer is impermeable to hydrogen ions. It's impermeable to any ions. They're charged. They can't get past. This is really critical. So now, we've got a set of proteins that are embedded in this membrane system. They're actually transmembrane proteins, and each one is made up of a couple of different proteins. And they're given the very clever names of complex 1, complex 2, complex 3, and Fred. No, it's complex 4. And then sometimes people will put in another complex, which I'm not saying I like to do. This is the hydrogen ion channel, and that's called complex 5. This is also known as the ATP synthase. Although, as far as I'm concerned, it is the ADP rephosphorylase, but whatever. All right, on your board exams, call it ATP synthase. I don't know. Now, here's what's going to happen. Let's just go ahead for the bacteria. We'll set it up like this. Here is the cytoplasm. This is inside the bacteria. And then this is the cell wall. And this is the great outdoors. This is known in the bacteria world as the periplasmic space. But we don't have to worry about that too much. Here, you can see that. Oh, there, periplasmic space. All right, so this is the setup. Now, the way it works is this. The Krebs cycle and glycolysis are happening here in the cytoplasm, and the electron carriers, NADH and NAD, they come here to be recycled. And the first thing that happens is NADH backs up here like a dump truck, dumps its electron onto complex 1. And it is recycled back to NAD. And it's given a little pat on the behind and told to go back out there and pick me up another electron. And it runs on its way. The electron is then jumping around complex 2, and it's going to complex 3. Now, there are actually a couple of other parts here, things that are called the cytochromes, and that's one of the terms of these different complexes as well. And there are in animals, and mitochondria and in eukaryotes, there are a bunch of different little vitamins, things like B12 are found in here. They are also kind of part of passing the electrons on. Let's not worry about that. All right, so the electron goes to complex 3. The electron then goes to complex 4. It's passed on like a hot potato. And then complex 4 is going to take the electron and dump it onto O2 to generate, just as we mentioned before, a molecule of water and the oxygen radical. All right, so now at this point, the electron has left the building. And if you think about this, we have taken the carbon from glucose, dumped that out as carbon dioxide. We've taken the electrons from the bonds and now dumped them onto oxygen, and it dumps out as water. It's just kind of bacteria pee or whatever. It just diffuses out. Now, the next little bits, we have a few details we have to pick up. One is, let's think about FADH2. FADH2 doesn't have quite the level of reducing power that NADH does. FADH2 fits in here. FADH2 is going to dump its electron right there. And, yeah, you can see that. It's recycled back to FAD. And it goes back out. Now, let's put in our hydrogen ions, our protons, all right? And red for protons. So complex 1 is pumping out a hydrogen ion. Complex 3 pumps out a hydrogen ion when it has an electron, and complex 4 pumps out a hydrogen ion. And what we got here then is a hydrogen ion gradient being generated. The pH ends up being higher. And remember, pH is the inverse log of the hydrogen ion concentration. The higher the pH, the lower the hydrogen ion concentration is. The pH will be like, it might be 8 inside the cell and 7 outside the cell. That's a 10-fold difference in hydrogen ion concentration. It's a pretty big hydrogen ion concentration. Now, the only place where hydrogen ions can make it back through is through the ATP synthase. It is basically a hydrogen ion or proton channel. Now, just as a little point, we can interchangeably use the terms hydrogen ion and proton. And it used to bug me, like, how can you say that? Remember, we talked a little bit about elements, and that an isotope is an element that doesn't have the same number of neutrons as it does protons. The most common form of hydrogen on the planet is what I would consider a isotope. Depending on your definition, it's not, but it is a hydrogen ion that doesn't have an electron, so it's positively charged, and it doesn't have a neutron. It simply is a single proton, just that one basketball sitting on the 50-yard line. So when we talk about hydrogen ions or protons, they are the same thing. A hydrogen ion is just a proton. So this is a proton channel. And this proton channel is a fairly complex set of proteins that as the hydrogen ion moves through, it is able to now rephosphorylate. We can think of it a little bit like a dam, where we have put all the water back here, and now this is the water inlet. We can let the water through and run a turbine. You know, theoretically we're spinning that. And what we're doing now is driving the rephosphorylation of ATP. So we've got adenosine diphosphate, and then we have a phosphate molecule floating around. This is being coupled together to form ATP. And this is where the bulk of the ATP is being generated. That is, we can say that we get 4 ATPs between glycolysis and the Krebs cycle. And if we have 36 total, that means we're getting 32 out of this system. And for all organisms, this is a bit of a weak spot. You can jack with this pretty seriously and screw animals and plants and bacteria up really seriously by monkeying around with this. One thing is, this system is dependent on having a perfectly intact and proton impermeable membrane. If we start to punch holes in this membrane, boot, boot, the protons can start zooming through here, not through the channel, not doing any work. They just come right back in. Now, there's some very cool stories with this. It's called uncoupling, and it actually is used, or one of the things that happens is the mitochondria in animals can generate heat this way. And if you're interested, holler, and I'll tell you some really cool stories about this. It's what's referred to as a thermogenic reaction. And it's not how you generate a fever entirely, but there are drugs that will actually punch holes in the mitochondria, and they will cause the body temperature to go up. Some interesting diet drugs, actually. It's a whole other story, but there you go. All right. Let's look to see what happens if we start jacking with this in some different ways. Now, obviously, if we've got drugs that punch holes in the membrane, that's going to screw things up. But what if we remove the oxygen from this system? What's the consequence of removing oxygen? Let's just go ahead and take a great big... Right here. Take a great big X and throw it down right there. There ain't no oxygen. So, no O2. What's going to happen? Watch what happens. It's not going to be pretty. With no O2 present, complex 4 has no place to dump the electron. And so it's standing there with its electron trying to dump it onto oxygen, and it's standing there looking like a dummy. It's like, no oxygen, I'm holding on to it, what do I do? Oh no. It just sits there. Now, complex 3 has an electron, and it says, okay, complex 4, here's my electron, take it. And of course, complex 4 is sitting there like a dummy going, oh, I got an electron, I can't take another. And complex 3 says, well, what the heck am I supposed to do? And then complex 4 goes, oh no. Now, complex 1 is ready to hand off its electron to complex 3, and it goes, okay, here you go. And complex 3 says, okay, I'm full. Complex 1 says, what am I supposed to do? And complex 3, of course, said the same thing that complex 4 said, oh no. And complex 1 now has got to talk to the customer. He's looking like a complete chump because he's got an electron. Here comes NADH backing up, ready to dump his electron. Ain't no room, can't take it. And NADH is saying, well, what the heck am I supposed to do? And it's told, sorry, you're going to have to come back tomorrow. And then NADH is going, I don't know what the heck to do. It just sits there. And within, about 30 seconds or so, all the NADH is all the electrons are there. There is no NAD left. The whole system grinds to a halt because you can't dump the electrons. And because you don't have any NAD, you can't run the Krebs cycle, which needs like 5 or so NADs per cycle. That can't go. And so pyruvate starts to back up because it can't dump into the Krebs cycle. That is a problem. And this is what's known as fermentation in this specific case. Now, what we're about to get into is some very cool twists on this whole play. And for me, honestly, it was one of these definitions that went, oh, I can make a distinction. Anaerobic respiration is when we put in something other than O2, sulfur, S2. That works great. Or iron, Fe2. That would work. That's anaerobic respiration. And so these are bacteria that are going to be obligate anaerobes. They are able to operate in a no oxygen environment, but they have to have their own terminal electron acceptor. Now, if you were to take a obligate anaerobe and not give it whatever it requires as its final electron acceptor, it's going to get screwed up too. But that's a different game. All right. So, let's think about a few twists and variations. One thing is, bacteria are fairly clever at figuring ways out. And this is an interesting bit that I want to just throw out and or throw up. And we'll pick up on Wednesday in just a moment. But let me just go through this here. This is an interesting little cut that bacteria can do. And let's look at it. It starts out way back here in glycolysis. And you guys are now all experts in glycolysis. You can write this whole thing down because you memorized this. Fine. Let's go through it. We got glucose coming in. And remember, we add 2 ATP to the system. It splits, it bifurcates into these 2 3-carbon molecules. And each one is making its way down. 3-carbon, 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 3-carbon. And this is pyruvate. And let's fill in. Now, It's one of the little reminders that I tell the guys that are studying for the MCAT, and I will tell you this as well. The way you can always keep up with the order of things is NAD is being used up and has an electron being dumped onto it ahead of the rephosphorylation of ADP. If it didn't happen that way, we wouldn't actually have to carry out this process. So let's look at this for a second. We've got NAD is being reduced to NADH on that side, and NAD is being reduced to NADH on this side. And then we have what's known in the biz as the ATP recovery step. ADP is being rephosphorylated to ATP. ADP is being rephosphorylated to ATP. And actually, there's two of them here. So this is how we get a little bit of ATP in glycolysis. But you got to have an NAD. Now, just imagine you're cranking along, but you run out of oxygen. You can't dump the electron that's on NADH onto oxygen because it ain't there. What do? Well, you could cry and close up shop, which is what I do. But there is actually a very clever little trick, and it's this. What if, rather than use O2 as the final electron acceptor, what if, and I'm making this quite verbose, what if we use pyruvate as the final electron acceptor? This is an interesting little question. So what we're going to do is not use oxygen as the final electron acceptor, but instead we're going to use a 3-carbon pyruvate. Now, this is what happens. We've got the two pyruvates here, so pyruvate and pyruvate. We're going to take pyruvate and we're going to add an electron to it. We're going to reduce it. So what we do is have NADH dump its electron onto pyruvate, come back out as NAD, and that can go right there. So we can just simply recycle this NADH and just bounce it right back up. And the same with this one. And now we've got the 3-carbon pyruvate. It has been reduced. It becomes a different molecule. And what it becomes is actually really cool. And a lot of divergence. There's a lot of different things it can become. Depending on the organism. You and I carry this reaction out. And we call this, in a sense, anaerobic respiration. But it's actually what is known as fermentation. So, if we use pyruvate as the final electron acceptor, the standard term for this is fermentation. And with animals and some bacteria, we generate a 3-carbon acid called lactic acid. And this, of course, is what contributes to muscle soreness after exercise. So you may not be thinking about it, but when you are exercising really hard, you actually begin to generate an oxygen demand greater than what your lungs can generate and what your oxygen can carry, your blood can carry. And you begin to ferment, particularly in the big muscles of your thighs, of your arms, wherever you're really exercising hard, you're using lots of oxygen, and you begin to ferment. It's also referred to as anaerobic respiration, but that's not really quite the right term. It really is fermentation. Now, with bacteria, this molecule ends up being a number of different things. It depends on the bacteria strain. It can be, like in animals, it can be lactic acid. It can also generate acetic acid and a molecule of CO2. We can generate ethanol and CO2. We can generate methanol and some kind of 2-carbon molecule. I'm not really exactly sure what. What you're doing is generating all these different kinds of products by reducing pyruvate. So it's not a very efficient way of operating. And there's some actual physiological consequences to this that, depending on the organism, can have some real serious consequences. If you think about us, for example, or mesophiles that are aerobic, when you're dumping that electron onto oxygen, you're making water. I mean, who doesn't love water? It's good. It's an easy, non-toxic metabolite. It is a waste product, but it's not something that's going to be toxic. So generating water, no problem. Now, if you are generating things like acids or alcohols, these actually can become a problem if they build up too much. Now, for you and I, if we begin to build up too much lactic acid, because we're carrying this part of the reaction out excessively, we can go into lactic acidosis, drop your blood pH, screw you up really bad. It's a fast way to either the emergency room or the grave. So it doesn't work real well. And let's think about what we're doing here. Rather than shutting up shop entirely, if there is no final oxygen electron acceptor, what we can do is this shortcut, and we generate a net gain of 2 ATP. So it is, for us, kind of a last-ditch effort to generate a little bit of ATP in the absence of oxygen. Now, for microorganisms, they actually can get by with this. It's not great, and it's going to slow their growth up dramatically. So if you can grow something like E. coli in an anaerobic environment, probably in your gut, that's how it's growing, and it'll generate these various acids and things, you're generating 2 ATP per glucose as opposed to 36 ATP per glucose. So you either have to eat a lot more glucose or you are going to be growing a lot slower. But that's okay. They're a single cell. They ain't got that much to do. So they're a cell. They sit there and accumulate ATP until finally they get enough that they can divide or something. And they do it. You and I, this really won't work very well and not for very long. If we're running on simply 2 ATP per glucose, that just isn't enough energy to actually carry out much in terms of metabolism. And so you end up building up lactic acid to a point that you can't survive. And so it is, for animals, really a last-ditch effort. For bacteria, it works. It just means that things are going to go way slower. But they grow in the absence of oxygen. Pretty damn cool. Yeah. All right. On Wednesday, we're going to pick up a little bit on some of the PowerPoint. I'm not sure if I posted it or not. I'm not sure I did. And this is a bit more of a generalized perspective of the metabolism, but I thought it's useful to go through this. You may be able to use this in A&P, in Essentials. It's really some good information that is either a review or a novel way of thinking about it, but there you go. Hey, thank you for your attention, and I'm getting on the exams. We'll be grading them. Hopefully by the end of the week we'll have it all. And that's it. I'm out. Peace.